All right, now we're going to do the neuromuscular junction. Basically where the neuron touches the muscle. Remember, it gets very, very, very close, but it doesn't touch it. We call this area the neuromuscular junction. Okay? So you have this neuron that's going to come all the way down here and touch the muscle. There's a gap in between here. Okay? Now, a synapse versus a neuromuscular junction. A synapse is a nerve going to a nerve, to another nerve, or a nerve going to a muscle. So that's what a synapse is. A nerve to a nerve or nerve to a muscle. A neuromuscular junction is specifically a nerve to a muscle. So all neuromuscular junctions are synapses, but not all synapses are neuromuscular junctions. All right, just a little nomenclature for you guys. Okay? And a synapse is going to be this microscopic space in between the neuron and the muscle, or the neuron and another neuron. Okay? And it's going to allow a message to go from the neuron to that. We'll be talking about the muscle, so I'll be using the word muscle instead of another neuron. So there's a chemical that comes off the neuron, goes across this gap, and goes to the motor end plate where the muscle is. That chemical is called the neurotransmitter. So let me show you what this is all about. All right? This is the axon, and this is the axon terminal. Okay? There's a gap in here called the synaptic cleft. And then this will be, let's say this is the motor end plate, which is really the sarcolemma of the muscle. So this neuron gets very, very close, but it doesn't touch. There's a gap there, a cleft. This, at the end of the axon terminal, we call this the presynaptic membrane. This over here on the motor end plate is going to be the postsynaptic membrane. Okay? And we're going to, if this is New Jersey and this is Pennsylvania, I know that is wrong geography, but just bear with me since I'm from New Jersey. I want New Jersey to be on this side. I couldn't find another picture that would reverse it. So you'll get the picture. So this is New Jersey, goes across the Delaware River to Pennsylvania. Okay? It goes across here. Now this is what's happening. An action potential comes down here. Goes to the axon terminal. Inside the synaptic cleft, there's calcium. Ooh, calcium's all over the place. So many more important things than making bones and teeth strong, right? Well, there's calcium that's in the Delaware River, in this synaptic cleft. And as the action potential comes down here, it's going to open up calcium channels and allow calcium to come into the axon terminal. When calcium comes inside here, it's going to bind on these black circles, which are vesicles. And inside the vesicles are these green dots that are neurotransmitters. In our case, we're going to be using acetylcholine as a neurotransmitter. So what happens here, as the action potential comes down, it's going to open up calcium channels, allow calcium to come into the cell, bind on the vesicles, which make the vesicles fuse with the presynaptic membrane, and through exocytosis is going to release the neurotransmitter to go across the Delaware River, or the synaptic cleft, until it reaches its receptors on the other side. Kind of like pushing a button. And it's going to start an action potential on this cell. And that's the part that we talked about with the neuromuscular junction. And we talked about the sarcomeres and all that stuff from yesterday. Okay? Does that make sense? All right? We'll go into much more detail than that, but that's very simplified. The skeletal muscle contraction. Okay. Contraction relaxation happens in 4 phases. Okay, and these 4 phases, when combined, is called a twitch. First thing is going to be excitation. The nerve action potential is going to cause the muscle to have an action potential. It's going to excite that muscle. So it's going to lead to, through that neuromuscular junction, the action potential of the nerve is going to make or lead to an action potential of the muscle by that neurotransmitter. Okay? Then you're going to have excitation and contraction coupling. The action potential on the sarcolemma then is going to activate the myofilaments, the actin and myosin. When that happens, when you got the bridging between the myosin and actin, you're then going to have that contraction happen. Okay? And then we have the point where there's going to be relaxation. That's the point where the resting, it's then going to relax. Calcium's going to go back into the sarcoplasmic reticulum. These things over here is going to be our twitch. And it doesn't twitch. There, that's what I'm trying to see. The extra side effect I want you to see. Okay. So that's what a twitch is. Now, what I have here for the next few slides, probably about a dozen slides or so, I think I have 3 videos I want to show you right now. And it's going to put all this in perspective. And I'm not going to go through each one of these different things, excitation, the different steps. But you can do this on your own. Like 3 goes with this, 4 goes with this. It's everything that we talked about so far, but in a little bit more detail. But the video is going to show it nicely. Okay? The only thing I want to just go over is one other thing. The axon and the axon terminal. The muscle. All right, I'm going to reiterate and try and do animation for you here, but you'll see the videos too. Here's the synaptic cleft, the presynaptic membrane, the postsynaptic membrane. Jersey, Delaware River, Pennsylvania. Okay? Inside the presynaptic membrane, or the axon terminal, you're going to have these vesicles that have a neurotransmitter. In our case, when we're dealing with neuromuscular junction, this is going to be acetylcholine. And we have acetylcholine receptors on top of this motor end plate. Make sense so far, what I'm drawing? The action potential is going to come down this axon. That's the axon potential. When it reaches the axon terminal, there's calcium out here. And when the action potential comes down here, it's going to open up calcium channels. And we have more calcium on the outside than on the inside. And the calcium is going to enter the cell. When a calcium comes inside, it's going to bind to the vesicles here. That's going to force the vesicles to fuse with the presynaptic membrane and through exocytosis is going to make the acetylcholine or neurotransmitter go across the synaptic cleft. It's going to bind to acetylcholine receptors over here. This is a gate that's going to open and allow sodium to come in here and cause the action potential to spread across the muscle. Depolarization, repolarization, and all. Remember, you have an action potential here in the neuron, but you've got an action potential here that'll go on the sarcolemma, down the T tubules, making the sarcoplasmic reticulum release the calcium to spread on the sarcomeres, exposing the myosin binding sites on actin, having the myosins bind to the actin, cause a contraction. Now that's all going to happen as long as acetylcholine is binding to receptors. But we have an issue. What if we don't want to keep on contracting, taking a glass and squeezing it until it breaks? So we don't need this acetylcholine to always be triggering that muscle to contract. So we got to get some of this acetylcholine off to allow us to relax. So we have, this is the part I didn't tell you about, we have an enzyme over here that's floating here. And this is called acetylcholine esterase, or cholinesterase. Everything's written on the slides over here. Acetylcholine esterase. Remember, if it ends with an ASC, it's an enzyme. ACH ACE. All right, you'll see my abbreviations up there. Okay. So what's going to happen here? It's also referred to as cholinesterase. Same thing. So you're going to have this floating around here, and it's going to take this acetylcholine, bring it in here, and break it up into two things. It's going to break it up, and part of it is going to be what we call choline. And choline is going to get absorbed back in here and get reused to make more acetylcholine in the cell. The other thing this is going to break up to is something called acetate. Or it's also called acetic acid. Vinegar. Well, acetate or acetic acid is all over your body. It's ubiquitous all over. So this acetate just will go and mix and mingle with the rest of it, which is all over the place. So that cholinesterase is going to take this and break it up. So that's what's going to actually not make this muscle continue to contract, because we have the acetylcholinesterase that's going to do this. Now, it could do it too quickly, but as long as there's an action potential going down here, it's always going to release more acetylcholine across here. Right? This is the acetylcholine. As As long as the action potential is going on, you're going to release more acetylcholine, allowing these buttons to be pushed so that you can continue contracting. Does that make sense? Okay? Let me show you the 3 videos, and it's putting all together now.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neural transmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine. Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers. The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads in thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross-bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
Let me see. Could I borrow you? I know. I'll show you something. And you don't mind if I touch your flesh on your arms, right? Okay. I got today's day and age. I gotta ask that. All right. So come on over here. I'm gonna show you something. A little more there, and face me, and put your arms out like this. Now, she's gonna be active. Okay. I'm gonna be myosin. Now, this is where the head of the myosin is, and I need to touch her flesh to bring her closer to me. I can't do that because she has a sleeve on. Okay? This is tropomyosin. Alright? Tropomyosin is this. And it's actually blocking my binding site on her arm. So what's going to happen is calcium is going to come over here. It's going to attach to something called troponin, which is going to expose. I'm not going to go that far. Okay. Okay. I'm going to expose her flesh here. Now what can happen is I can actually bind to here. So my myosin hand is going to bind to her actin, where that binding site is for myosin. I can bind here, and I'm going to do this kind of movement. Release, recock, and do the whole thing. So it'll be like this, go like this, bring it closer like this, and so forth. And she's getting closer to me. Does that make sense? The actin is not getting shorter. The myosin is not getting shorter. It's just that we're getting closer because they're overriding each other. As long as calcium is all over here, this sleeve is going to be pushed up and it's going to expose the myosin binding site so I can grab onto. I can't grab onto the sleeve. Once the calcium goes back in the endoplasmic reticulum, this sleeve is going to go back over here, and now I can't bind here, and that causes relaxation. Took me a while to figure out that little analogy. Does that help out? Okay, thank you very much. Okay. So does that make sense about actin and myosin and the cross-bridging when they touch each other, bringing it over here? All right, calcium is important to move that sleeve away from the binding sites. All right, once we have no calcium, you're going to have relaxation. Okay? So, in summary of the skeletal muscle contraction, all right, again, what we had up here, because I want you to really know what's going on here. That third exam, a lot of it's about action potential, a lot of it's about neuromuscular junction. I'm going to show you how you can apply it with a lot of diseases. Okay? The action potential comes down here on the axon. Okay? It's going to allow the calcium to come into the axon terminal. The calcium is going to bind to the vesicles, allowing the acetylcholine to get released through exocytosis and bind across over here to its receptors. This is then going to allow sodium to go in, potassium to go out, and it's going to actually make it more positive inside to a negative 55, which means now the muscle is going to have an action potential. And that's going to go down the sarcolemma, down the T tubules, allow the calcium to come out of the sarcoplasmic reticulum to cascade what I put with her arm, and so that the sleeve goes up and my myosin goes on here and cause a contraction. How they came up and found out about this is people who have no social life. All right, but it does all these different steps. Understanding all these different steps allows us to understand a lot about diseases and how we can find medications or treatments for diseases, understanding these little fine points. So that's steps 1 through 6. And if you look over here on what I drew on the board in this is 1, 2, 3, 4, 5, 6. It's doing the same steps that you just saw before. The only one that's not up there is 7, which is acetylcholine esterase, which is going to take acetylcholine and break it up into acetate and choline. Remember, acetate is also known as acetic acid. Same thing, vinegar. Okay? And that's what acetylcholine esterase is going to do. Calcium is then going to be released from the sarcoplasmic reticulum into the cytoplasm. Calcium binds to troponin, what I did with her sleeve. Okay? It's going to expose the myosin binding site on actin so that myosin can bind to actin and cause the contraction to occur. ATP is needed to break that connection between the myosin and actin so that we could actually have another power stroke. ATP is needed to break it up again and do another power stroke to have her come closer and closer to me. Okay? This is from the physiology coloring book, and it kind of puts things in perspective. This one here, and I have another one in the next slide or so. Okay, so you could read this over here if you want. It's doing that whole thing. The steps. The T tubules going over here. There's two terminal cisternae over here. What it all does. All right. Again, showing the same thing that I just did with the sleeve with her. Okay. So the sliding filament hypothesis is basically placing your right. I read this someplace. Taking your right palm on the back of your left hand. Doing that. And what's going to happen here, if you now slide the right palm towards your left elbow, what happens to the distance between your elbows? They got closer, right? They got shorter. We're not changing the length of the arm or the length of this arm. It's just they're riding over each other. And that's what we call a contraction. Myosin and actin, when they're riding over. It's more or less like this, though. It's kind of like doing this kind of crawl. Because it has to come up, power stroke, do this way. And ATP is needed to break that bond. All right, so they slide over, makes a Z disc, which are basically my elbows get closer together. And then what we have is it's going to pull the bone towards wherever you need it to do. And voila, you have movement. So it does that. They ride over each other. The microfilaments don't get shorter. They just overlap each other, and they do that. And this is where all that medical alphabet we talked about. Here's a relaxed, and here's a contracted. You can see the H zone over here. It's pretty wide, and it's pretty short here. Remember the two zones that do change is the H zone and the I band. Okay? I band over here, see how wide it is? But then with the contraction, it got shorter. So you can utilize this to understand why or how the I band and the H zone change, whereas nothing else changes. And the sarcomere itself, from Z disc to Z disc. So there you have one. That's one sarcomere. That's a Z disc to a Z disc. Doesn't really do much work with one sarcomere. You put 3 there, it's going to do a little bit more. Remember, a microfilament has about 10,000 sarcomeres on it, so they get more and more. And when you get further away from this, you get to start visualizing and seeing the orientation or the arrangement causing the striations in skeletal muscle. It's because of the way the actin and myosin are arranged that create those parallel lines, the striations in skeletal muscle. Okay? So I got a question. What causes rigor mortis? If you don't know what rigor mortis is, when someone dies and they're left there in the bed, and it only happened like a few hours ago, they will be stiff. Their muscles are fully contracted, and it's difficult to break that contraction. So why is it when someone dies, they get rigor mortis? Yeah. That's part of it, but we got to go into the cell. And remember how we said myosin and actin are bound there? What's going to release the myosin? ATP. And what do you need to make ATP? What 2 ingredients? Glucose and oxygen. So if someone's dead, are they breathing? So they're not making ATP. So this is going to stay contracted. Acetylcholinesterase is another thing, but that happens later on because the enzymes are still there. It takes some time for them to break down, but you can't make ATP. For your body to make 1 round of ATP, you'll be able to survive 1 minute. ATP is very important for you to survive. You need to keep on making ATP even when you're sleeping. Okay? So that's what's happening over here is there's no ATP to break that bond, or that bridging is what we call cross bridging. So you start having this sarcoplasmic reticulum releasing calcium, which causes the muscle to contract. But death, there's no oxygen, so you can't make ATP, and ATP is required to break that myosin bridge from actin. This usually happens within a few hours, 5 to 6 hours or so. This is when it starts kicking in, and it stays like that. And it'll stay like that for depending on the person, but it stays like that up until about just under a week. But it could last only just one day, until the muscle tissue starts breaking down itself. There's no nutrients there, so they start breaking down, and then it just loosens. That's why when forensics, they go and they could have an estimate of when this person died, because they can see how long if rigor mortis didn't set in yet, they know it happened, a few hours ago. Okay, and that's what a rigor mortis is. It's hard for me to find a picture with someone who's got rigor mortis, so I'm sorry for putting such a gruesome picture here. But basically, a guy who committed suicide, he had this shotgun in his hand, and when he finished it, his arm stood where the shotgun was. Does that make sense? All right. Again, sorry it's gruesome, but I don't know how else to show you what rigor mortis is. There are some people that don't know what rigor mortis is, and it's that. Okay.